I am wearing two hats today, and I will be telling you about two different data resources. And one of those my team runs, and the other one I have got slides from the team leads in charge of the resource. But rather than just do the two talks in succession, I've merged a little bit. So I'll give you some of the background to what we're doing overall at EBI, and then tell you about each of the specific resources, and I'll probably pause midway through just to catch up on questions, and then finish telling you a bit about where we are going with both of those, because I think that will work better than just single resource talk. So the introduction, I work at, and in fact sitting in the buildings of EMBL-EBI, and we do a lot of things around biomolecular data. And we are part of the wider EMBL, the European Molecular Biology Laboratory, and we are one of the 6 sites of EMBL in 5 of our member state countries. And EBI, the European Bioinformatics Institute, is the one that's situated in the UK, and we do purely computational science. So there's somewhere between 600 and 800 people, depending on who you count, all working on different aspects of data. And that involves some training, it involves research, but what EBI is best known for is that we run scientific services. And what scientific services really means is databases, data resources, so archives, knowledge bases. And these are covering the whole range of different data types that we work with in modern biological research, from chemical data through to gene expressions, sequences, literature. But what I will be telling you about specifically is the imaging resources, because imaging has become more and more and more important as a computational data type over recent years. And that's why workshops like this are so important, that we manage that imaging data effectively. And imaging at EBI in some senses fits into the wider context of EMBL's program, because EMBL as a whole does a lot of work in imaging at very different scales. And in fact, this slide comes from the EMBL program, showing the different scientific services actually in physical imaging across length scales from the atomic through to the organism level. And because imaging is really a whole family of different techniques, it's not just one thing, there's lots of different bars crossing different scales. But what I want to show is that we offer resources that cross all of these. And really, these began out of the PDB, the Protein Data Bank, and that's not an imaging resource. That provides protein structures, or more accurately, models of protein structures. It's the oldest resource in computational biology. It's more than 50 years old now. But when the cryo-EM revolution took off and we started to derive structures from really images rather than X-ray crystallography, that gave rise to a need from the community to store the raw images that underlie electron density maps that give structures. And that gave birth to the first imaging resource at EMBL, and that's EMPIAR, the Electron Microscopy Public Image Archive. And although EMPIAR started in the world really of structural biology, it's since expanded to other forms of 3-dimensional electron microscopy, so in particular cryo-electron tomography and volume EM. But that's still quite a narrow part of the space of all of the imaging that we do. What about all the length scales up here, for one thing? So in 2019, EBI launched the BioImage Archive, and the BioImage Archive in some sense has 2 roles, one of which is to take the rest of the data types, and actually that's most of them, because light microscopy is still by far the most common type of imaging that we see, but also to provide, in the longer run, an overarching sort of single point of reference for both deposition and access to types of imaging data, possibly in the long run, not so much the structural data, but the rest of the data that crosses the length scales. What I won't talk about now, because Jean-Remy will talk about later, is another extremely important part of the imaging data ecosystem, the IDR, the Image Data Resource, which holds very comprehensively annotated reference data sets. And I won't mention that because Jean-Remy will cover it. But what these resources are there to do is provide routine deposition as deposition databases for all of these different types of imaging across all of the scales, really from the near atomic, so there's a tomogram up here, it's a little bit hard to see probably at that resolution, through to tissue and up to organ and organism scale. And not only images, but also annotations, so labels and segmentations, because those are incredibly important when it comes to really understanding images. What's a deposition database? A deposition database, and we use that phrase quite a bit at EBI because we have lots of data resources and they tend to be classified into deposition databases and things that build on them. A deposition database takes data, and typically it takes data at the time of publication. So usually a scientist has done some research, or more often a team of scientists, they've done research, they are publishing a paper. Increasingly, we are all expected to make data open at point of publication. And when we do that, the data is deposited in a deposition database. And that's usually a combination of the data itself, but in this case, images and some minimal metadata. So as well as told you about metadata, I'll touch on it again briefly in the deposition database. We take that and we give it back out again, and we give it out again for lots of different reasons. Often the most immediate reuse of data is for methods development. So most commonly in image analysis, actually very AI driven these days, but also for training and for teaching. But in the long run, for de novo scientific discovery, and the resources that have run for a long time have shown that there's lots of really interesting things that can come out of lots of well-structured data at scale. The other set of use cases that I won't talk about so much are that the archives power added value databases, knowledge bases, things that can take the data, they can put perhaps an alternative front end on as a data portal, combine data from different domains, they can reprocess the data, they can add extra information, they can be knowledge bases. So somewhat different use case, but also a really important one. So the two EBI deposition databases, EMPIAR and the BioImage Archive, fit together to cross these scales. As I mentioned, at the moment, the route is that EMPIAR takes structural biology, so cryo-EM and cryo-ET data, and three-dimensional electron microscopy, and the other things, so two-dimensional EM light microscopy, go into the BioImage Archive, although some of that may change in the long run, and I will touch on that right at the end of the talk. When it comes to deposition, then you will be guided through this process. So we have much more detailed information about exactly what the scope of the different resources are on the help pages. So you don't really necessarily have to think about this until it comes to the time to deposit data. What I would say overall is that we can take the data no matter what the imaging modality is. The only restriction that we have is that we don't take patient identifiable medical data. The focus is really on the sort of, we do have data sets which are human data sets as long as they're anonymized, but we're not really focused on the preclinical side. It's before that, it's the more bioimaging research. So the first of the two resources I will tell you about is EMPIAR, the Electron Microscopy Public Image Archive, and this is the talk I'm giving on behalf of the team that run EMPIAR. And as I said in the overview slides, EMPIAR really grew out of structural biology. So structural biology has a long history of very community standard archiving starting out of the protein data bank. And if you're interested in this, you can go and read more about it, and you can go and find out both the history and exactly where the different resources, where the different types of data as part of structural biology go. I'm assuming in general that this audience is not so much, because coming in by Euro-imaging, it's not so much a structural biology background. So I will focus on really on the latter part of this, where EMPIAR has expanded to include volume EM and X-ray imaging data. So the most EMPIAR entries are, as I said, structure determination. They look a little bit like this. There are lots of series of images together with pointers towards the structures that derive from those images. And those structures sit in the information about those structures actually sit in the EMDB, the electron microscopy data bank, with experimental information in the protein data bank, which is where pigeon 3D models like the one on the right in the tweet show. But the part of data that's probably most relevant and useful for this audience is three-dimensional EM data. So EMPIAR has a growing collection of data from the various serial sectioning or ion beam milling electron microscopy techniques, and those include quite a range of things. So, example on the left is plant data, the examples of, as I mentioned, human data, which is in a sense biomedical in use, but not patient identifiable. So EMPIAR is still dominated by cryo and cryo-ET. That's about 90% of the data. But as I mentioned, there's a growing set of these other different types of information, particularly the structural biology focus, the non-structural biology, the 3D EM, and also some soft and hard X-ray tomography. EMPIAR has been growing pretty rapidly since it started in about 2013. It hit the 1 petabyte milestone in 2021, but it hit the 4 petabyte milestone earlier this year, and that's pretty rapid growth. It's maybe not quite exponential, but close to it. So it's really filling up a need in the community, and actually this follows the fact that protein structures are increasing, or deposition of protein structures, I should say, is also increasing exponentially. I won't go into a huge amount of detail as to the exact mechanics of the deposition processes for both resources, but it's good to give you a high-level overview for if you do come to deposit data. EMPIAR, I guess before I get onto deposition, I think I've spent even less time on this search and data access, because although that's incredibly important, that's not so much the target of the talk today. Suffice to say, perhaps, that there's a good search system. You can come, you can find the data, you can access the data via a number of different transfer protocols. They're more oriented towards large bulk data transfer for EMPIAR, so things like ASPR and Globus, because that's what you typically need if you're getting very big data sets to do reprocessing of structural biology data. There is also pretty extensive help for both access and downloads, and that is in the form of the web pages. There are deposition manuals. There is a YouTube channel with talks and tutorials. The high-level picture of the way that deposition works, and actually this is the same for both of the data resources, is that you register yourself, because although data access is completely anonymized, for depositing data, we need to know who you are, so you can go back to the same deposition, among other things. So you register, then you go through some web forms, and I'll show you a screenshot of one of those in a moment, to give an overview of the deposition. You upload the data, and there's a couple of different, in fact 3 different ways to do it. Again, mostly oriented towards bulk data transfer, because these data sets tend to be very large, and then you provide a little bit of extra information about the individual image data sets. And that takes place all through a web interface, which will guide you through the relevant steps and let you fill in all of the data. You will find, when you do this, that because of its history, the current EMPIAR deposition interface is quite oriented towards the world of small scale things in structural biology. So for example, it will ask you for the spacing between your image planes if you have 3-dimensional EM data in angstroms, because that's the way the structural biologists think in angstroms. And that will probably change a little bit in the future as we make it more general. I mentioned there's different ways to upload files. ASPR and Globus are probably a little bit less familiar unless you've worked with big data beforehand, but they're much better for very large data transfer than more traditional things like FTP, because FTP is part of the scale. I'll stop there for EMPIAR. So turning now towards the BioImage Archive. So this is the resource that is probably more generally relevant, just because volume EM data is still a smaller component of the community, and actually this would cover the question so far. The archive takes a very, very wide range of data. So I should have delayed our image when I'd shown this slide. We have, this is a small subset of thumbnails and images that we hold, and it includes lots of fluorescence microscopy, as you can see, and there's light sheeting among those. There is whole slide imaging, histopathology. There are 2-dimensional EM images. There are slightly more unusual things like shortwave infrared. And actually, this one over here, this slide, this is part of the one of those biodiversity museum collections, as I said. So it does include this large scale macroscopic organism scale, not traditional microscopy. As well as the data being very general in terms of modality, it's very general in terms of the research topic. So often it comes from areas of human study, like say immunology. So I think this central image is humans. I think it's thymus, perhaps, but actually other model species as well. So I think this one's pig and this one's mouse. But then much more sort of pure fundamentals research. So this is zebrafish from developmental biology, effectively, and then this is, I think, fission yeast, maybe, looking at cell shape control. So lots and lots of very different things. It is, I think this is relatively unique about the BioImage Archive, in some ways completely unique globally, is that it is a deposition database that will take your imaging data more or less no matter what you're doing outside the medical space. The archive started, as I mentioned, in 2019. The reason this graph starts a little bit before 2019 is because we took in some data from a resource that sits underneath the BioImage Archive called BioStudies. Things really kicked off here, and actually we didn't have a dedicated team to run the archive until 2021, which is when I started. So most of the active development has happened in the last 2 to 3 years. And in those 2 to 3 years, it has grown very rapidly indeed. Again, this is not, if you sort of do the model fitting, it's not exponential, but it's more than linear. As of, I think, this morning, when I updated this slide, there are a little bit over 600 data sets, lots of different imaging modalities, counting by the ontology that we use. And at the moment, we get about 20 submissions a month. Projecting forward, the sort of major milestones in terms of incoming data we'd expect to hit soon are that later this year, it'll be on average, I think, about 1 submission a day that we process, and probably the 1,000th entry sometime earliest next year. But actually, this year things are increasing even faster, I think, than the projections had indicated. We follow a set of our metadata model. So the information we capture follows a set of guidelines called REMBI. So REMBI is actually really a set of instructions for making metadata models. It sort of tells you what kind of information you should capture. And we, about 18 months ago, implemented in our submission system capture for most of the components of part of this model. And I'll show you what that looks like in practice for an entry in a moment. But the intention here is not only do we capture the information relevant to the image, but we can link to other forms of information that, depending on the type of study that the image represents, may be relevant. So, for example, for compound screens, we would link or encourage linking towards or chemistry databases, and so on. I won't, again, because the focus here is more about both overviewing the deposition process, spend a lot of time on how you access the data, but the front end of the archive will let you search. There's quite a good search system which is aware of synonyms and things like that, and it'll let you browse and filter. Again, I'll talk right at the end about what we're working on to make that even better. But when you see each of the submissions, you will see a page that looks like this, which is called a landing page, and this summarizes the information. I'll take you through what's in one of those landing pages, and I should flag that this is a really nice submission. A lot of the reason this is such a nice submission is because Isabel was a data steward for this submission, so she spent a lot of time helping the team submit the data, and I think this highlights why the role of data steward is so important in the long run in making our data effective for reuse. So the information that in the REMBI model we would call study information, you might sometimes call that kind of investigation, well, somewhere between investigation and study in the ISA model. That information is really background context. So it's, for example, a link to the relevant publication. It's an explanation of what the data set represents. It's the people that were involved in both the production of the data, but also the curation of the data, importantly, where Isabel is listed, keywords, the license. Importantly, or this well, and unfortunately, I'm not quite sure, this will tend to come up. We also capture quite a bit of information about funding if you choose to give us that information. And part of the reason for that is that funders increasingly are quite specific about exactly how funding should be acknowledged in data deposition. So there's both flexibility here, but also guidance as to what you should include, for example, in EU-funded projects where they want project acronyms and streams and things like that. The information in some sense for reuse gets a little bit more interesting when you start to look at the next stages, and those are what we call the biosample, which is information about what the actual thing that was being imaged, what organism does it come from, what tissue type is it, if it's a cell line, is the cell line, is it modified, is it a genetic knockout, for example, and then specific information which is about how it was prepared for imaging, so what was necessary to do in order to be able to get it inside the microscope. And then for each of the images in the study, there's tabular information, which is the information that's relevant at the individual image layer. So for example, if magnification differs across the images, this is the information that will vary from study to study because different things are relevant in this case, because this is human samples. It's important to know the stage of the donor, for example, but for a compound screen, this would have the links to the chemical compounds. The way the images are presented in this tabular format also lets you download both all of the data, but you can filter by that information. So for example, if you just wanted images associated with a single compound in a compound screen, or in the previous case with a particular donor, you could do that. And we have the bulk download options from EMPIAR. The EMPIAR also has the FTP and Globus and Aspera for then big studies. And we have a few more options for small data sets because the light microscopy tends to also produce a lot of very small data sets as well as the really big ones you see more on the EM side. To just about on time, high level, again, overview of the submission process. It is mostly very similar to the way that EMPIAR submission works, in that you will look to make sure you're depositing to the right place, you will register, you will transfer data over, and you will provide information, that metadata, that can be metadata, or most of that can be metadata that accompanies the images. The thing that's a little bit different about the submission mechanism here is we have this thing we call a file list. And what a file list is, let's skip forward a couple of slides because that's what I'm talking about now. This tells you, or this tells us really, and therefore the downstream reusers of the data, both what the individual image files in the submission are, but probably most critically, what the individual image level metadata that's relevant to those images are. So in the examples, in the example I showed, it was that information about the donors. This is the information that would be, again, compound screens, it would be chemical compounds, genetic knockout, it would be some identifier for the gene and so on. Because this varies a lot depending on the type of study, we provide guidance and templates rather than specify an exact set of things that must be included. Over time, we're probably likely to need to introduce more validation for this, just to make sure that the keys and values are more controlled. Back briefly to the data transfer, you upload the data. We again provide these bulk data transfer options, FTP and Aspera, actually not yet Globus, although we're working on that. For the BioImage Archive, you can also upload the data just through the web interface. You don't have to use a command line client. Again, because there are a lot of light microscopy data sets can sometimes be quite small, sort of, a few gigabytes maybe. And for that, something that small, it's easier to do it through the web interface. When you get very large data sets, you tend to have to use the bulk data transfer. The accompanying metadata goes in through a web form. The one thing I wanted to highlight here is that when you select where you're submitting, almost all of the time, there's an option at the top where you choose the data type, which is just the BioImages Archive study. But we do have a specialized submission route, and this is fairly new as of a couple of months ago, dedicated purely to annotations. So if you're just submitting, for example, segmentations or point annotations or something like that, which is most relevant usually if you're doing some kind of machine learning, then there's a route for that as well. So this is what the form looks like. It will let you type things in. It will validate at a reasonably basic level what you type in. I guess the last thing I'll say on submissions specifically and comparing the submission types. Where the BioImage Archive and EMPIAR differ is that EMPIAR has an internal what's called annotation route. So when you fill the form in, it passes to a human who will review and communicate with you about what's happening in that submission. The BioImage Archive actually doesn't do that. All of the validation is automated. So as soon as you submit, then the data, if you want it released immediately, will be released, depending a little bit on transfer time. And there's no human in the loop review, and that's actually really necessary because of the kind of scaling we're expecting in the future. But it's something to be aware of if you weren't expecting necessarily the data to go live immediately. There's lots of help for the process. There's also a useful online video that will guide you through at a high level, and then there's varying levels of detail down to, I think, what we're seeing here, which is the study metadata. Things that tend to matter that I'll say this again right at the end when I give the summary. When you finished, data get copied to permanent storage. You can choose when the data will be made public, though. So once you've given it to us, it isn't made public until the point at which you decide. And the reason for that is that often you want to time that process to coincide with the release of your manuscript or maybe your preprint, but you want to do the upload in advance so that the timing can be right for releasing. As soon as you finish the upload, you will get the accession identifier and the DOI. Just be a bit careful because the DOI will point to something saying you can't see that yet, or we don't know about this thing until you've made it public. So we give the DOI in advance, but just be a little bit careful about the release date. Release dates can also be changed. So you'll specify it when you complete the submission, but you can push it into the future. And again, because of review processes, sometimes you want to do that. I will run so we don't go too far over very quickly through things that we have done very recently and we're doing. A lot of the focus in the moment or recent focus has been on this route for deposition and annotations. The other thing that we've been doing, and I'll show you very briefly in a second, is actually getting some in-browser visualization of the data, because I've sort of shown you pictures, but actually you wouldn't see those when you look at the individual studies. So the reason annotations are important is really for the AI community, and that's where what we've done over the last year, a little bit longer than a year, is get some community consultation on what metadata standards specifically for annotations should be. We've implemented these in the deposition system, and now we have a route where data can go in along with annotations or with annotations separately, and that gives you data that's kind of ready for AI model training. And there's now a section of the web page where you can access these specific data sets. The last thing I'll show just before far future, and this is something that I think we put in last October, is that for now a subset of our data, this is not yet all of the data. Scaling this up is the thing we're working on. We have in-browser visualization for the images, and this is using the fantastic developments on file formats and visualization tools linked to those around OME NGFF, the new sort of cloud-ready file format, which you'll hear more about later in Bura's talk. And we're doing that both for the image data from the BioImage Archive, and we're actually doing it for the image data from EMPIAR as well. So the previous example was histopathology. This is volumetric electron microscopy. Viewers differ a little bit in each case because the kind of things you want to see differ depending on the data. If it's very big 2D data, you want the more sort of Google Maps-like viewer. If it's 3D data, you want the orthographic projections. I won't mention 3D rendering for the moment. This is sort of really cool if you can do it for CT data. It's perhaps more specific to certain data types. Last things, 1 minute past time, but I don't go past 1 minute. We are working on updating the web page because it's a little bit out of date. This is a strong area of focus for the moment. We are also working on search that is aware of images rather than whole studies. So if you search at the moment, you get information that's relevant to all of the images or very specific metadata. We're working on search that really combines both the data from EMPIAR and the BioImage Archive as the first step towards much tighter integration between those resources, but also is giving you back images together with relevant information that we harvest from the images, and that harvesting via primarily the bioformats to raw, so the OME tool feeds into that search system. The other thing, and again, I won't talk about this, it's probably relevant to a smaller group of the community, is we're looking at how programmatic submission would work. So I will stop there. Summary, headlines. We can take the data for routine deposition across different types of imaging data that are not medical. Don't worry too much about the fact that the submission process went by very quickly. There's good guidance through the process. Doesn't cost you anything. You get back the bits you need to put in the publication, and you can control the timing for those publications. Actually, you could also provide reviewers and editors with private access to the data, which again is something that's often journals will want through the review process. So I should thank my team as well. I should also thank the EMPIAR team, and I won't pass their slide very quickly.